All right, we're going to pick up here talking about some of the mechanics and actual physiology of sound waves and how we actually engage in this process of hearing. So, there are a couple of terms that we need to be familiar with, all right? And these are amplitude and pitch of the sound, all right, or frequency. So, when we're talking about the amplitude, so the amplitude has to do with you can think of it as the height of the sound wave or the vertical distance from the valley to the peak of an individual sound wave. So if we're looking here, if we do this in something like blue, we have this bottom part of a sound wave, and then we have a top part of a sound wave, right? So the distance from this bottom part, so from this valley, all the way up to this peak, that is known as the amplitude. Okay, so this is the amplitude of a sound wave. The loudness of a sound is completely dependent on the amplitude. So the greater the amplitude, the greater the loudness. All right, and this is measured in decibels. So you may have heard of the term decibels. This is sometimes recorded in sporting stadiums that record the loudness of an event. So this has to do with how tall, essentially, these sound waves are. So this sound wave here, if we're drawing something like this sound wave would not be as loud as a sound wave that looks like this, right? So it's just not the same, all right? Whenever we're talking about pitch, this has to do with the frequency of the sound waves, all right? So the frequency has to do with the horizontal distance from one peak to the next peak or from one valley to the next valley, right? So from this point here over to this point here, that is frequency, right? You could also do it from the peak, right? From this point here over to this point here, that is the frequency. Frequencies that are shorter, so the greater the frequency or the higher the frequency, the higher the pitch of the sound. The more spread out or the lower the frequency, the more spread out it is, the lower the pitch of the sound. Okay, so we've all heard high-pitched sounds, high-pitched squeals, screams, whatever, and we've also heard very, very low-pitched sounds as well. Okay, all right. So when we're looking at this, this is going to show the pathway of sound from the outer ear to the middle ear, and then we'll look at a separate slide here in a little while that is showing the pathway of sound from the middle ear to the inner ear, right? So we've already talked about this a little bit kind of indirectly as we've gone through talking about the different features of the outer and middle ear. So sound waves are obviously going to start in our environment and be funneled in via the auricle or the pinna, right? So you have the best ability or the greatest ability to hear things most clearly and get the most direct transmission of sound waves when the sound waves are going directly into the ear, right? So say the sound wave's coming directly from here and it goes straight through the ear canal toward the tympanic membrane. You have a lesser ability to hear, or your ability to hear things is slightly decreased when the sound wave is coming from an indirect position. So imagine you are covering your left ear, and so you only have your right ear, say, available for hearing, but the sound wave is coming from the left, right? So if we have this ear, imagine the sound wave is coming from over here. It's got to come and it's got to be bent and it's got to be distorted, and then it can enter this ear canal. So you're obviously going to lose some of the amplitude, some of the loudness, and the clarity of this sound wave, okay? All right, it'll then enter into that auditory canal, right? So it's going to enter into your ear canal here. We can then strike our tympanic membrane, right? The tympanic membrane will vibrate, it will move. When it moves, it causes a sequence of movements in those auditory ossicles. So first moving the malleus, moving the incus, and then moving the stapes, right? So malleus means hammer, the first one, incus means anvil in the middle, and then the stapes is that third one that is then pushing on the oval window. So where we end, when we get to our middle ear, is we're ending with the pushing of the stapes, the foot plate of the stapes, on the oval window that then transmits the sound into what is known as the vestibular duct. And then we'll see here, there's a membrane down here known as the basilar membrane that we'll look at in a second. The sound waves here, depending on the frequency of the sound, whether it's high frequency or low frequency, it will distort and move this basilar membrane either very, very proximally, so very close to the oval window, or very far away from the oval window. Okay, and that's, again, that is dependent on the frequency of the sound wave, all right? So once we move past the outer and middle ear, we get into a region known as the inner ear, all right? And so the inner ear is going to be involved, or the cochlea is going to be involved with our inner ear. So whenever we look here, this whole structure, all right in here, this is all our inner ear, specifically associated with the our sense of hearing. So we have another part of our inner ear that's involved with our sense of balance and equilibrium. All right. So within this ear, you may have heard that we have fluid in our head. We have fluid in our ear, and that's true. We have fluid in these ducts, in these ducts within our ear. All right. So the two fluid types are what are known as endolymph and perilymph. All right. Endolymph is going to be within a region known as the cochlear duct, and the cochlear duct is going to be this kind of blue, this blue duct that we see right here. This is going to be the cochlear duct. And then there's going to be two other ducts known as the vestibular duct and known as the tympanic duct. So the vestibular duct is going to be this one right here, where we just drew this arrow, and the tympanic duct is going to be this green one right down here. So in the cochlear duct, we're going to have endolymph. All right, endolymph is going to be rich in potassium. And so if you recall, when we did cells, when we've talked about cells before, the intracellular fluid is very rich in potassium, right, and not very rich in sodium. So when we look at endolymph, think of endolymph as being similar to the intracellular fluid in that it is high in potassium. And again, it's in this cochlear duct, so where we're talking about right here. The perilymph is similar to the extracellular fluid. So we said extracellular fluid in our cells was very rich in sodium, right? So very rich in sodium, and this is where we have this perilymph in our vestibular duct here, and in our tympanic duct down here, right? And again, this is going to be very rich in sodium. So as that stapes is pushing on that oval window, the movement of the oval window is going to distort the fluid in this vestibular duct. It's then going to go across and deflect the basilar membrane, which is right down here along the bottom of this cochlear duct. And then we'll see that the deflection of the basilar membrane actually has implications for moving what are called hair cells, and these hair cells are going to result in us actually being able to hear sound. Okay. And then down here we have a round window. So if I actually clear this so it's easier to see, the round window is going to be in this area right here, and this is serving as like a pressure relief valve. So as we push the fluid here, it gets distorted across this membrane, and then it comes out and exits and bulges out at this round window right here. So it serves somewhat as a kind of as a pressure relief valve. All right. So when we're looking at our cochlea, the cochlea is this kind of spiraled, snail-shaped organ in our inner ear, and it contains something known as the organ of Corti, sometimes known as the spiral organ or the spiral organ of Corti. All right. This organ of Corti is essentially kind of this whole structure that we see up here. This whole thing essentially is the spiral organ of Corti. All right. So what it's doing is it is converting the pressure waves that are ultimately coming from the tympanic membrane, from the malleus, the incus, and the stapes through the fluid, and they're converting that movement of those bones, movement of the fluid, and movement of these hair cells that we see right here. They're converting that into a nerve impulse. So they're converting that into an action potential or a membrane potential. All right. This organ, the spiral organ, is within our cochlear duct. Right. So this cochlear duct that we see right here, this organ, the spiral organ of Corti, is found within that duct and surrounded by that endolymph fluid that we just talked about, right? So here's our endolymph, and again, here is the deflection of this membrane that we'll discuss here in just a brief moment, okay? So what happens is we have, if we kind of trace this movement of sound waves again, we go from the external environment, hit the tympanic membrane, move the malleus, incus, and stapes. Stapes pushes on the oval window. Now we're in the inner ear, all right? As that foot plate of the stapes pushes on the oval window, it's moving the vestibular duct, okay? Now another term for this vestibular duct, you may have seen this in a few of the other slides, is the scala vestibuli, okay? This also is the same thing as the vestibular duct. It's this superior duct that you see right here. This bottom one, the tympanic duct, is sometimes known as the scala tympani, all right? So as we push that stapes, the foot plate of the stapes, it pushes on the oval window and it bulges the fluid and moves the fluid within this scala vestibuli. All right. Now, as you move the fluid within the scala vestibuli, you have it start to travel along that vestibular duct, and then at some point it can deflect across what's known as the basilar membrane. All right. So the basilar membrane is this more inferiorly located membrane on the bottom of the organ of Corti. So it's this kind of purple membrane that you see stretching right here. This is the basilar membrane. All right. And so as the foot plate of the stapes is pushing on the oval window, so imagine this structure down here. If you look at this kind of wave generator in this bottom right corner, imagine this machine that's pushing here, that's going in this direction here. Imagine this is the foot plate of the stapes, and it pushes on the oval window, and you get these fluid pressure waves, this distortion of the fluid within the vestibular duct. So at some point, depending on the frequency of the sound wave, this signal, this movement of fluid is going to deflect, if we look up here in the top left, deflect across and cause movement of this basilar membrane. Okay? As this basilar membrane moves, it is going to move these hair cells against this structure right here, which is known as the tectorial membrane. All right? And we'll talk about that in a little more detail in just a second. But that's kind of an introduction. So think about this movement of the fluid. The vestibular duct comes across, will move this basilar membrane, and that's going to move these hair cells that we'll talk about in just a second. Okay. So if we go here, this is talking about movement of the oval window is transforming and transmitting those vibrations through the vestibular duct as these waves of pressure, like we see here in the right. Okay, in the right picture. So as we do this, we're going to have this pressure, these pressure changes. They're going to be transmitted to the cochlear duct. Okay, so remember, the cochlear duct is in the middle between the vestibular duct and the tympanic duct. All right, so the vestibular duct here, this is in blue. So as these pressure changes are transmitted to the cochlear duct by movements of the basilar membrane, so the basilar membrane, as we said, it's this layer that we see right here. As we move the cochlear duct, right, as we distort the fluid there, we're going to move the fluid within the cochlear duct, which remember, that is called endolymph, and it's going to distort this basilar membrane. As you distort the basilar membrane, it moves, and it kind of shears these hair cells that we see right here. It's going to shear these hair cells against the structure known as the tectorial membrane. All right. So imagine you have you have one hand and you have your fingers up against the palm of your other hand, and as you move your fingers, they're kind of being distorted and they're being pulled against the palm of your hand. Well, that's essentially what we have here. As this basilar, if we're looking at this image up here, as the basilar membrane moves, it's pulling these hair cells that we see right here, and it's shearing them against this tectorial membrane. And we'll see in a second that there are little, what are called stereocilia, these little kind of hair cells, these little fibers that stick up. And as the cells move, those stereocilia actually get pulled in one direction. And if they get pulled in one direction, you get depolarization. And if they get pulled in another direction, you get hyperpolarization, or you don't get depolarization. Okay. All right. So this displacement of the basilar membrane is related to the frequency of the sound waves, as we said. So what this means is that high-frequency sound waves are going to distort the basilar membrane at a different location than low-frequency sound waves. And it turns out that based on the shape and the size and the flexibility of the basilar membrane, high-frequency vibration or high-frequency sound waves distort the basilar membrane closer to the oval window, and low-frequency sound waves distort the basilar membrane further from the oval window. Okay, and we will look at a picture here that makes this make a little bit more sense here. Okay, so here we see this transmission of sound waves. So again, we go from the outer ear to the inner ear, tympanic membrane, malleus incus stapes. Stapes pushes on the oval window, and that's where we start right here. So we're pushing on the oval window, and we have fluid moving in our vestibular duct. At a certain point, the fluid, the pressure waves in this vestibular duct is going to be transmitted and transferred across the cochlear duct. And as this happens, it's going to move the endolymph, it's going to move and distort the basilar membrane, it's going to pull those hair cells against the tectorial membrane. That's going to initiate an action potential. All right. So as we've mentioned a few times, this distortion of the basilar membrane is going to be dependent on the frequency of the sound wave. All right. So if we look here, I'm going to come back to this in just a second because we've kind of already mentioned it. Here's our basilar membrane. This is what we're looking at in these images right here. Depending on what the frequency of the sound wave is, we're going to move the membrane at a certain point. So if we're moving it, so here's our stapes. So this is the oval window. You can see we've distorted the membrane at this point here, very, very close to this oval membrane. So this means we must have a low frequency, or I'm sorry, high frequency. Let me turn this into an H. This is a high frequency sound wave, all right? This one down here, you can notice here's the stapes again, here's the foot plate on the oval window, and this is where we have distorted the basilar membrane. So this is going to be a low frequency sound wave. And it turns out that the thickness and the size and the flexibility of the basilar membrane changes from where it is near the oval window to where it is further away from the oval window, okay? So let me go back and we'll talk about this slide that we skipped here. So this is showing the transmission of sound waves now in the form of pressure waves and fluid movement within our inner ear. So we go from the oval window, which again, we're starting right here. Here's our oval window with the stapes. It's going to move the fluid in our vestibular duct, right? So we've done this one. We've done this one. We're moving fluid within this vestibular duct. At a certain point in this vestibular membrane, we're going to move the fluid now within this cochlear duct, which is this middle one that we see right here, right? So we've moved the vestibular membrane and we're moving the cochlear duct. And the base of this cochlear membrane, we have the basilar membrane, or this cochlear duct, we have the basilar membrane. We will then distort that. That's going to move the hair cells, and the pressure wave will be transmitted to our tympanic duct that we see right down in here. And then the pressure waves will be kind of relieved at the round window where it bulges out kind of right there, okay? So we've done that one. So this is a very, if you haven't noticed, this is a very three-dimensional, very visual process. And so sometimes looking at two-dimensional images like this, even though they're numbered and you can see the flow throughout, sometimes it still doesn't really make a whole lot of sense. And so if you watch a video or you watch an animation that shows these events occurring, movement of the fluid, the perilymph in the vestibular duct, deflecting across the cochlear duct, moving the basilar membrane, moving the hair cells, all this, it'll probably make a little bit more sense, okay? So we've talked about this here. And so here we see the same thing. Depending on the frequency, so here we have very high frequency, right? Here we have very low frequency. We're gonna move the basilar membrane at a certain point. So high frequency near this oval window, and then low frequency, we're moving it further away from the oval window. So that's what this slide is showing, high frequency, and then low frequency, again, is moving it further away. So we can have this frequency tuning of the cochlea depending on the frequency of the sound wave, okay? So here's the same image that we saw earlier, high frequency right here, close to the oval window, low frequency further away from the oval window at a more distant location, okay? So what we see here is this is the actual mechanism by which we get an action potential that is ultimately going to be sent to the brain. So we have that, if we go back a few slides here, let me go back to one that'll make a little bit more sense. So here we're in our vestibular duct, right? We have sound waves coming, and depending on the frequency of the sound, as we just said, it's going to deflect the basilar membrane at a different point. So say it then dives, or we'll just continue down to where we're at right here. It's going to deflect across. Okay, so 4. This is number 4. If we read number 4 down here, it's saying pressure waves cause the vestibular membrane to move, resulting in pressure formation in the endolymph. Remember that endolymph is this fluid within the cochlear duct, okay? This is then going to move this region of the basilar membrane that we find right here, which is then going to distort those hair cells against the tectorial membrane, okay? So that's what we see happening in this image right here. So if we go back to our slide, we'll see that movement of the hair cells due to the movement of the basilar membrane pulling these hair cells, so these stereocilia here, and this one large one called the kinocilium, moving these against the tectorial membrane is going to cause them to open ion channels if they're pulled in one direction, and movement to close the channels if they move in the opposite direction, right? So if we move in the direction of the kinocilium, this kinocilium, this is the longest stereocilia, essentially. So it's this largest one that we see right here. Okay, that's the kinocilium. If we move in that direction, then we're going to open our ion channels. When we open ion channels, remember, what are we going to have out here? We're going to have lots of potassium in the endolymph. That potassium is going to flow in. When we bring in a positively charged ion, we know we're going to depolarize the cell, right? So we brought in cations, calcium, or potassium, we depolarize the cell, and then we have a neurotransmitter release. So that's what's happening right here, right? So whenever we release our neurotransmitter glutamate, we're then going to stimulate these postsynaptic neurons that are then going to go, this is part of our cochlear nerve, and it's going to send the signal back to the central nervous system, back to the brain, all right? So this is what happens whenever we depolarize, right? We're depolarizing the cell, we're actually getting an auditory stimulus, and we hear, right? So whenever whenever the deflection of the basilar membrane in this endolymph pushes these cilia away from the kinocilium, right? So away from this one large stereocilia here, our ion channels are going to close. When they close, we no longer have potassium rushing in, right? So we no longer have this potassium rushing in. So we get repolarization, and glutamate is no longer going to be released. So you see down here, we no longer have glutamate being released at our synapse. So we no longer have this transmission of the action potential toward the central nervous system, toward the brain. Okay. So one important concept of all of this is how do we get, how are we getting this nice complex perception of sound from simple sound waves, and how can we amplify it? Well, one good way is that we take the sound waves that strike our tympanic membrane, which is relatively large when we compare it to our oval window. So we have this large surface area, right, of our tympanic membrane. We have this large area that is transmitting a lot of force onto the malleus. So this is the low pressure, so right, very large area, low pressure. We're transmitting that same pressure or that same force onto a very small area. So now if you have a small area, right, small area, you have a very high pressure, right? So these are inversely related. So you have this large tympanic membrane, and through the action of the malleus and the stapes, you apply that force over this very, very small area of the oval window. So this allows us to amplify the sound wave a very great deal in order to have that sound wave be amplified heading toward the inner ear and then ultimately toward the brain. Okay. So as we age and as we have certain things happen in our environment, we could potentially damage our hair cells. So if we damage our hair cells, they're not very capable of regeneration. So this damage is largely going to be irreversible damage due to loud noise. And so we talked about earlier this idea of these two muscles in the ear, one called the tensor tympani, one called the stapedius. And these are thought to help to dampen these extremely loud sounds and help to kind of decrease the activity of the tympanic membrane and decrease the activity of the stapes in an attempt to prevent these things from happening. So to prevent all of the sound wave from coming into the inner ear, and you have this rapid movement of these hair cells, and then you ultimately have damage of those hair cells as well. Okay. All right. So we're going to stop here with our hearing, with the audition part, and then we will pick up talking about equilibrium in the next part, still staying in the ear.